Welcome to our practical class on estimation of photosynthesis using IRGA, that is infrared gas analyzer. This is the picture showing the process of photosynthesis. Photosynthesis takes place in a plant organ called chloroplast. Plant draws up water through its root. Leaves takes carbon dioxide from the air. It traps energy from the sunlight, and the plant uses energy of the sunlight to turn water as well as the carbon dioxide into sugar and oxygen. Oxygen will be released from the plant part, and the plant uses the sugar. Carbon fixation or carbon assimilation is the process by which inorganic carbon is converted to organic molecule. The most common method of carbon fixation is photosynthesis. During the process of photosynthesis, carbon is fixed, that is, it converts from inorganic to organic form. From the carbon dioxide assimilation rate, photosynthesis can be measured using an apparatus called IRGA. This is IRGA or infrared gas analyzer. This helps in the measurement of instantaneous net carbon dioxide assimilation rate in an. Let's see the principle behind the infrared gas analyzer. Infrared gas analyzers are used for the measurement of a wide range of heteroatomic gas molecules, including carbon dioxide, water, ammonia, carbon monoxide, sulfur dioxide, nitrous oxide, or nitric oxide, and also the gaseous hydrocarbons like methane. Heteroatomic molecules have a characteristic absorption spectrum in the infrared region of the electromagnetic spectrum. This is a picture that's showing infrared region of the electromagnetic spectrum. That is, these heteroatomic molecules, they absorb this region of infrared region of the electromagnetic spectrum. Therefore, the absorption of radiation by a specific heteroatomic molecule is directly proportional to its concentration in an air sample. That is, say for example, in the case of carbon dioxide, when carbon dioxide concentration increases in an air sample, higher is the absorption of infrared radiation. Also, when carbon dioxide concentration reduces, the lesser the absorption of infrared radiation. Infrared gas analyzers measure the reduction in transmission of infrared wave bands caused by the presence of gas between the radiation source and a detector. The reduction in transmission is a function of concentration of the gas. The primary role of IRGA is to measure the carbon dioxide concentration. The IRGA is sensitive to detect even a change of 1 ppm of carbon dioxide. The leaf or a plant is enclosed in an airtight chamber, and the carbon dioxide flux determined by measuring the carbon dioxide concentration changes in this chamber atmosphere. This is the airtight chamber in which we keep the plant or leaf for determining the photosynthesis. In this slide, the first picture that's showing the infrared absorption spectrum of carbon dioxide. Carbon dioxide, which is an heteroatomic molecule that absorbs infrared radiations. The major absorption peak of carbon dioxide is at 4.25 micrometer, with secondary peak at 2.66 and 2.77 and 14.99 micrometers. And this is another graph that's showing the absorption spectrum of carbon dioxide as well as water vapor. Both these molecules absorb infrared radiation. So infrared radiation in 2.75 micrometer range. LICOR 6400 is an open system for laboratory or field photosynthesis measurement of leaves. It has two main components, the console and the sensor head. Console is the part where the values are calculated. It has a display and a keypad. On the right hand side of the console, there are sensor head connectors. These are sensor head connectors that connect the console with the sensor head. On the lower side, there are battery compartments, and this is the battery that we are using in IRGA. On the left hand side of the console, there are carbon dioxide scrubber, desiccant tube, and a carbon dioxide source or tank to provide carbon dioxide to the IRGA. We can also attach field stands to the underside of the console. Then comes the sensor head or IRGA. It has leaf chamber. This is the leaf chamber where the leaf is placed. And there is also a handle. It is connected to two Peltier thermocouples, two Peltier thermoelectric coolers. And then we have sample and reference gas analyzers. There are two gas analyzers, infrared gas analyzers on the sensor head. For the light measurement, an external quantum sensor is connected. One quantum sensor is also connected with the sensor head for measuring the light. And for measuring the leaf temperature, a thermocouple is held at the bottom of the leaf chamber. This picture showing the sensor head of the IRGA. The incoming air stream passes through two gas analyzers in the sensor head, which allows obtaining absolute carbon dioxide and water values as well as from the reference as from the sample IRGA. The basic principle is that carbon dioxide is a strong absorbent of infrared radiation. So an infrared source shines through a sample chamber and onto a detector. The energy at the detector is the total energy entering in the system minus that absorbed in the sample chamber. One pair of carbon dioxide and water analyzer defined as a reference IRGA that measures the carbon dioxide and water vapor concentration in the ambient air that is sent into the leaf chamber. Similarly, a second pair, that is the analysis or sample IRGA, that measures the carbon dioxide and water vapor concentration in the air that is coming from the leaf chamber. The difference between the reference and the analysis IRGA or the sample IRGA are computed. A leaf is clamped in the leaf chamber. The leaf chamber is provided with suitable pads to clamp an area of 2.5 cm square under airtight conditions. Separate tubing is provided to send and withdraw air from the leaf chamber. These tubes are connected to either of the reference or analysis IRGA for the determination of gas concentrations. A quantum sensor is placed inside the leaf chamber's transparent cover to measure the actual light intensity of photosynthetically active radiation range at the leaf surface. The quantum sensor is also shown on the right hand side of the slide. Blue and red LED lights is fixed on the top of the leaf chamber. LEDs emits light in the PAR region, that is photosynthetically active region, and the intensity of which can be fixed and controlled at a required level. The light source is capable of providing the photosynthetically active radiation in the range of 0 to 2000 micromoles per meter square per second. A carbon dioxide cartridge normally carrying 8 grams of pure carbon dioxide in liquid form is used to get the requisite carbon dioxide concentration in the leaf chamber. The system mixes the ambient air with the carbon dioxide to obtain the requisite Concentration in the leaf chamber. The path of the ambient air is provided with two scrubbers to remove the moisture and also the carbon dioxide. To remove moisture, it is a diurite used as a desiccant, and for the removal of carbon dioxide, it is soda lime is used. Let's see the working procedure of IRGA. First, charge the batteries 1 day prior to record data. Load the charged batteries first. Then connect the carbon dioxide tube to the inlet of the instrument. All the screws of this instrument must be in tight fitting. Connect the carbon dioxide tube in a proper way. Connect this tube very tightly, otherwise it shows leakage in the display. The second edge of this tube was kept in empty thermocol box or enclosed for uniform entry of the air into the tube. The next step of working IRGA, that is switching on the instrument. When we switch on the instrument, the display shows a welcome to loading open system. We need to press enter. Then the display shows is the chamber IRGA connected Y or yes. If it is connected with the console, there is IRGA connected with the console, we have to press yes, that is the letter Y. Then open the IRGA leaf chamber one time and close it. Then select new measurements. For that we need to press F4. In the display, select open log file. For that, press F1. Give file number, name and press enter. Next step is matching IRGA. For carbon dioxide matching, select match, that is pressing F5. Wait up to we get equal values of reference carbon dioxide and sample carbon dioxide. If we need to close matching, press match IRGA, that is F5. After that, press exit. For that, we need to press F1. So in this condition, we need to wait for 15 to 20 minutes for warming up the instrument. That is before inserting the leaf in the IRGA chamber. And it should be remembered that leaf should not fold in the IRGA chamber. If leaf gets folds, it shows negative reading. Leaf should not have any moisture and dust before inserting the leaf. Let's see what are the parameters that we can record it from IRGA. The first and the most important thing, that is the photosynthetic rate, then stomatal conductance. It is represented by CONT, which can be seen in the display. Then transpiration rate, it is TRM mole. Then intercellular carbon dioxide concentration, it is represented as CI. Then chlorophyll fluorescence,